Hello, students. Welcome to week 6 of the Next Generation Sequence Analysis class. This week, we'll be continuing our discussion on the resequencing pipeline and the methodologies that are commonly applied to calling SNPs in next generation sequencing data. I guess I'll start by reviewing just quickly what we talked about in the last lecture from week 5, where I gave you an outline of the steps to conduct the alignment and alignment processing, which are necessary in order to obtain the highest quality SNP call set. So the essential steps here are laid out on this slide. The main points being that we align the reads using a short read aligner like BWA. We have some details here to help to clean up the data. This realignment step around insertions, deletions is a very important step necessary to reduce systematic artifacts in the alignment process around insertions and deletions. But today I'll sort of describe the steps involved in SNP calling after these sort of alignment processing steps are completed, and then also describe some of the advanced topics related to obtaining the high quality SNPs, particularly in human data sets where the resources are available to call SNPs. I'll also talk for a while about calling SNPs in non-human data sets, which introduces a number of significant challenges, which I'll describe. So this is a figure that I wanted to review going all the way back to week 1. This is from a Nature Genetics review, or Nature Review Genetics rather, from Rasmus Nielsen and colleagues. And some of this now should look a little bit more familiar to you because this is sort of a generalized version of the previous slide. One of the things I wanted to point out is that SNP calling in large sequence data sets is typically done with what's called multi-sample calling. Okay, this is where you actually take multiple BAM files, each from different individuals. You typically merge those BAMs into one large BAM consisting of many individuals. Each read is still tracked, and it's known which read comes from each individual, obviously. But this multi-sample SNP calling is sort of the industry standard now. You typically, when you have a number of individuals from the same population, call SNPs across all of them at once. And we do that for reasons I won't get into detail about, other than to say that this helps to reduce the false positive rate. However, in your homework, so that's sort of the left track here in green. Whereas in your homework for week 6, I have you do what's called single sample SNP calling, and we do that here just to keep things simple. I'll have you calling SNPs in the same BAM file that you were working with in the SAM tools exercise from week 5. But I do really want you to know that about this multi-sample SNP calling, and that if you had, say, 100 individuals in a data set, you wouldn't sort of run the pipeline 100 different times for each sample. You would actually just run it once with a merged BAM file incorporating multiple individuals. Okay, and then this last step here has to do with some of the SNP steps I'll be talking about later, including the SNP filtering and the sort of advanced topic of variant or genotype quality score recalibration. So just kind of a reminder of what we're talking about when we talk about variants. So there's multiple different types of sequence variation that we work with in resequencing data sets. Single nucleotide polymorphisms illustrated here, sort of in this cartoon here, where we have this might be an individual that differs, that's a homozygous, say, that in other words has two alleles, one from each parent for the G allele, whereas the reference genome has a single A individual, implying that this individual is homozygous for A. There's also what we call small insertion deletion polymorphisms. In case if you've forgotten or never knew, this is a visualization of individual reads here, similar to the SNP example. But here, the black, heavy black line indicates missing bases. Okay, these are bases that are present in the reference that are missing from these reads in the sample. And so, if the reference essentially has what we call the insertion allele here, where the samples have deletion allele. So just as an aside, when we say indel, insertion deletion, what we mean is that we don't know, which individual had the mutation event that's derived or non-ancestral. Okay, so we don't know if this is a deletion that's occurring in this sample or if it was an insertion in the history of the allele that's represented in the reference genome. So we just call it an insertion deletion, simply to indicate that the two are different relative to each other in terms of a length polymorphism. Finally, we have structural variants. We'll get to talk about these hopefully a little bit in week 7. And these are different types of structural variants that we'll be talking about. So I wanted to clarify a point that's, I think, not obvious to people, and that's the difference between what's called variant calling versus genotyping. So a variant is a SNP. Okay, so this simply means that a particular position in the genome is variable in the population of individuals that have been sequenced. Okay, so it simply means that somewhere out there are at least two alleles in your sample that distinguish one or more individual from the reference. So what is genotyping then? Well, genotyping is actually the assignment of a genotype to an individual sample. Okay, so one is a variant is in the context of a population. Variant calling is the process of inferring that there is a site that's polymorphic. Genotyping is then the separate process of assigning an individual genotype to each sample in your data set. So this is actually something that I talk about in some other talks that I give about why do we just need to know variants? Well, because usually we want to know the genotype. That's true in many population surveys. All we are most interested in is estimating the allele frequency, which we can oftentimes assess without actually calling genotypes. So the problem with calling genotypes ultimately is that you need a lot higher coverage data than you do just to say whether a site is polymorphic, i.e. that it's a variant in a population. Genotyping requires deeper coverage, oftentimes above 10x, 15, 20x, or higher are frequently referred to as higher coverage, and therefore we can get more confident genotypes. So again, this is what real data might look like, where you have a site that's polymorphic. And I just want to introduce the word potentially here, because as I'll come back to later, while this looks like a polymorphic site, there's a lot of machine artifacts, alignment artifacts, and problems that can make a site that looks like it's polymorphic actually not be. And so I'll be coming back to that later. But briefly, I just wanted to tell you about some of the SNP calling methods that are out there, and where historically we simply used what were called consensus methods. This is just sort of base counting. So in the previous example, there's both A's and T's at this variable position. So we might infer that there's a AT heterozygote in this individual sample. And that may well be true, but modern SNP callers take into account the base qualities of these T's and A's at this position and incorporate that base calling error probability into a SNP calling and genotyping mathematical model. So these are probabilistic methods that I'm calling here. And they use a technique for parameter estimation in statistics called maximum likelihood that allows you to estimate the probability of a genotype given the data that are available to you. And ultimately, all I really wanted to say here is that these methods provide measures of uncertainty, and they naturally incorporate both the read depth and base qualities in assigning those error probabilities, whereas sort of these consensus methods approach essentially, you don't have those error probabilities in the variant calling or in the genotype assignments like you do with these probabilistic methods. And this is just to reinforce that there's a lot of math that goes into this. This is well beyond the scope of this class. But in multi-sample SNP calling, we have sort of two phases. We have, the phase that determines the likelihood of each of the three genotypes. In the case where you have two bases at a particular position, and then the second phase of sort of combining across samples in this multi-sample SNP calling approach that's now sort of the industry standard in large resequencing data sets. So in the end, all of this mathematical inference allows you to simultaneously infer, both the most likely genotypes for each of the samples, as well as their respective error probabilities, in addition to returning an error probability that there's a variant at that site at all. And hopefully some of this will become a little bit more clear as we actually look at some of the raw outputs, where we get both the qualities for a variant or a SNP and the qualities for the different genotypes. So I just thought I'd touch briefly, because if some of you go on in clinical settings or in inferring SNPs in human data sets, the human resequencing world is far more advanced at the moment than any of the other organisms that are studied. And the reason for that ultimately is that humans have a very large data set from the HapMap project and other genotyping projects from the mid-2000s that are very high quality SNP data sets. These serve as benchmarks which can then be used to evaluate empirically evaluate the errors that may be common in these short read resequencing data sets. And one of the things that came out of having those true positive data sets is realizing that the error probabilities are actually wrong. So remember, we've talked a lot about Fred qualities and how in your raw FASTQ data we have individual bases each have a Fred quality score, which can be converted to the probability of that individual base being an error or called incorrectly by the algorithm that processes the raw data coming off of, say, an Illumina or a solid sequencer. And so this slide here is from a paper, Mark DiPristo. This is a leader of the Genome Analysis Toolkit team at the Broad at MIT in Cambridge. I don't really have, I'm not really going to try to attempt to explain this slide, but I have posted this paper on the course site for week 6 because I think it's a landmark paper in our field, particularly in the resequencing field of next generation sequence analysis. But essentially, here are the observed sort of base qualities that are reported by a machine. Okay, so technically it's the empirical minus the reported quality. But so this deviation away from this blue line indicates the extent to which the error probabilities that are reported by the machine are incorrect. And this is the dinucleotide base context in this particular figure here for an Illumina machine. These he evaluated different sequencers and different sequencing contexts. Okay, so this is machine cycle here. So what this indicates is that in purple, okay, is indicating that at different positions in the read, the Illumina machine sort of the bias is varies along the read. That's particularly dramatic in the 454 where you see this funny shaped curve. The point is here that the base qualities coming out of the box after they come off the Illumina are not calibrated appropriately. Okay, and those biases vary based upon different features of the data, like their sequence context and their machine cycle. And the important thing to know is that in order to come up with high quality data, we need to adjust those error probabilities. Why is that? The reason is because that genotyping likelihood model that I just showed in the previous slide relies on those error probabilities. And so if those error probabilities are, say, systematically overestimated, as they appear to be based upon this figure here, okay, that means that you have too much confidence in a particular read, in a base at a particular position in a read. So if you have too much confidence, then what that can lead you to do is incorrectly infer that a SNP is there when in fact it's not there, because it's, say, a base is in error, but you don't think it's an error simply because the error probability is low, as predicted by these algorithms from each of these individual sequencing companies. So that's a long way of saying that we need to do base quality recalibration. Again, this is kind of review now because I've described much of this. The error probabilities coming off the machine, that is the Fred quality scores in those FASTQs, may be inaccurate. In fact, the previous slide shows that nearly always they are, and they are frequently overestimated. In other words, your confidence is overestimated. So why is this important? Because the genotype likelihood models use those error probabilities. We need a large database of SNPs in order to do the recalibration. These are some details that I won't go into because this is really an advanced topic. It's something that we've worked with extensively at NYU and tried to build into our pipelines. Although I will add the caveat here that for non-human data, this doesn't work very well, simply because we don't have this large database of SNPs, which is necessary to come up with the empirical estimates of the error probabilities. So these are advanced topics. I do not expect you to know in detail. I just throw this in here and spend 5 minutes or so on it because for those of you who will go on to clinical settings or on to large, say, bioinformatics or sequencing facilities, this may be common pipeline material if you're working in human genetics. So this is just to show you how that the recalibration actually has effects on the base qualities. So these are these FASTQC plots that I've shown you a couple of, and here these are Fred quality scores on the y-axis. This is the machine cycle or the position in the read, which is reflects the original machine cycle. So position 1, the first cycle of the sequencing run through. This looks like a 75 base pair reads. So here you notice how these box plots, the red lines here, the median qualities of the bases across the entire FASTQ, and you can see that the median qualities here in the before calibration are higher than they are compared with the after recalibration. And so this is the basis for statements like I made previously that we have evidence that these basis qualities are initially overestimated. So another advanced topic, and again the same reason why I presented the base quality recalibration is I present this for the same reason. Variant quality recalibration is also now a technique used in the human literature. The reason again being that well, in this case, we have what we call systematic machine artifacts, and there are artifacts after the SNP calling step that can be detected using machine learning. So essentially, machine learning, if you have a true positive set of SNPs with a set of characteristics, their sequence contexts, their Fred quality, base qualities, the variant qualities, along with many other features of those SNPs, including, say, their GC content, where they are, the extent to which the reads that support a particular SNP are biased on one strand of the DNA or the other. These types of information all go into a large machine learning algorithm that essentially allows researchers to detect sets of variants that look to deviate in certain properties from sort of normal SNPs. And these, this is just kind of a visualization of that, is that these are, you end up with sort of clouds of points that have, normal sort of signatures, and then, you have clouds of points that deviate from those, and those likely represent errors. So, again, this is something restricted to the human literature, and the reason you can do this machine learning is because you have true positives. In the end, if you've done any of the information processing sciences, you know about things like precision recall or ROC, or the receiver operator curves. These are techniques that allow you to sort of evaluate the specificity and sensitivity of a particular data set and make informed decisions about which sets of SNPs to keep and which sets to reject based upon some, say, false positive rate that you're interested in maintaining. This is sophisticated. Once again, this was a landmark study by DePristo et al. These methods are all applied in the genome analysis toolkit, which we'll be talking about later in the lecture, and you'll be working with in the class. We aren't going to incorporate these last two steps in your resequencing pipeline simply because of the complexity, and you need to be working on very large data sets and very large numbers of samples in order to get these methods to work anyway. And so for working on just your home laptop computer, these are fairly computationally intensive methods which we won't have time or the resources to apply in the course. So again, if you move on in human genetics, I want you to be familiar with the variant quality recalibration step. So let's move on, and I talked and showed you this slide a minute ago about a sample potentially having differences from a reference. So I say potentially simply because there's lots of reasons why a site like this might not actually be a real SNP. And one of the things about these mathematical models that take the error probabilities from read data and assign genotypes and can assign then a probability of that genotype being real or false, those genotyping models all assume that the read mapping, that is the alignment step, is perfectly correct. Okay, so in that there is no error in the mapping. However, we know from real data that there are lots of problems with the mapping. And so that leads to this topic that I want you to be aware of, and that is the basic idea that there are lots of potential pitfalls and errors in these resequencing data sets, and we need to be aware of them to have the highest confidence in our SNP call sets. So one of the things about error in these data sets is that they will be study specific. So how you go about preparing your libraries in the laboratory, the depth of coverage, which has to do with oftentimes how much can you afford, how many lanes of sequence data can you collect for a particular individual, and then post-processing, like your alignment processing steps that I described in the last lecture, and as well as these steps like the base quality recalibration, etc. These things will all affect the error structure in your data. So this statement here, I'll come back to in a second, really oftentimes, if we don't have the nice data sets that you have working with humans, we're left with doing a lot of filtering. Okay, so we're taking sets of SNPs, we're trying to identify characteristics of those sets of SNPs which look to be problematic. So how do we go about doing that? Ultimately, this is one of the problems in inferring where the errors are in your data. One thing about error is that most studies, they try to reduce the false positive rate. So you try to identify classes of SNPs, say ones that are covered with very few reads. Those ones are probably enriched for false positives compared to, say, high depth positions in a sequence genome. So, and with this error in these data sets, this is a review point. We can't utilize these well-known data analysis techniques to adopt certain specificity and sensitivity criteria. So how do we go about doing it then? How do we identify sets of SNPs that seem to meet some quality threshold to make us comfortable that we're not misinferring SNPs in the genome? Ultimately, this has some level of arbitrariness in it if you aren't using these methodologies, and what we end up doing is using things like the transition-transversion ratio to try to identify classes of SNPs that are problematic. So what are the sources of error? Well, the reference genome is not perfect, so you can infer SNPs because the reference genome itself has a miscall. Those are probably quite rare. The reason being that the reference genomes are typically based on Sanger sequencing, which is we consider to be the gold standard for limiting the false positive rate in DNA sequencing. There are errors introduced during library preparation. An example of this are the PCR errors I referred to in the lecture. They're the base calling errors. This can be due to a lot of imaging problems or chemistry problems during the sequencing run itself. And then there are read mapping errors. And I wanted to say something here because I think that which of these last two are major sources of error that we try to deal with in our data analysis. And which of these are relatively more important in causing errors will depend on the coverage depth. So base calling errors, if you have really like a lot of reads at a particular site in a particular sample, okay, the chances that the base calling error rate, let's say it's between 1 and 5%. That is, between 1 and 5 reads will actually have the wrong base. 1 and 5% of the reads will have, on average, the wrong base at a particular position, simply because of a base calling error during the sequencing. Or the processing of the sequencing data by the machine. So your FASTQ has errors in it. So, but if you have 100, reads, and you only have, say, 5, because you had a 5% error rate, where at a particular position, you have an error, then those errant reads are going to be at such low depth at a particular position that these genotype likelihood models, these mathematical models that infer the genotype, are usually not going to get it wrong. Okay, so you're unlikely at high coverage to suffer from these base calling error problems. The read mapping errors, however, are constant across coverage depth, okay, because the read mappers are simply aligning the reads, and there's a lot of reasons why they would align them incorrectly, which could lead to spurious SNPs. And so that's sort of independent of the coverage depth. So in high coverage data, the problems are mostly going to be due to read mapping errors, not base calling errors, whereas at low coverage data, both are contributing to the error. So let's look at a type of read mapping error that causes a problem. It's a common one. Let's imagine that you have what we call a paralogous region. So this is a duplicated region in the sample genome relative to the reference. So this fragment here does not exist in the reference genome. Either it's been deleted or this piece has been duplicated in the sample. We don't know, and we don't care. But whether it's a duplication or a deletion in which sample or reference. But the point is that relative to each other, they're different. And imagine a slightly diverged paralogous region or a slightly diverged duplicate. In this case, let's imagine this is a SNP. And if we go ahead and then sequence the sample and map the reads back to the reference, again, this is a mutation differentiating these two regions, then all of these reads from both of these two regions sequenced in the sample will map then to the same position in the reference genome. Okay, and what does this look like? If this is a single mutation, this looks like a heterozygous SNP in the sample, and that would lead to a SNP calling error. Okay, that's a common type. I've told you about misalignments around indels and illustrated an example in the last lecture. We have multiple mappings, so this is cases where a read is sequenced, represents the sequence of a region that's found multiple times in the sample genome and multiple times in the reference. So the read can map to any of many places in the genome. That can be problematic. And then here's this problem of mapping bias favoring reference alleles over non-reference alleles. I'll come back to this in a second, but imagine if you had a heterozygote individual that you've sequenced, and one of the two alleles in the heterozygote is the same allele that's in the reference. Okay, so that allele is inherently going to map better to the reference than will the alternate allele. The reason being because that allele has the same exact sequence as the reference, whereas the non-reference allele has differences, one or more, could be several differences, and therefore is less likely to map to the reference simply because it may not match optimally at that locus and may match somewhere else because it's different. So that's another class of read mapping errors. And I just wanted to reiterate that presently these mathematical models that infer the genotypes and the probability of there being a SNP at a particular position in the genome are essentially assuming that the mapping errors are non-existent. And so those error probabilities in the mappings are simply not incorporated into the SNP calling models simply because that would be an extraordinary complex thing to incorporate into the models. So how do we, in the end, get rid of these alignment errors? Well, we do a variety of things. The first things I've told you about here, we use realignment around short insertions deletions. So after doing the initial BWA alignment, we then come in and do the indel realignment step, which I'll expect you to incorporate in your resequencing pipeline. This I won't be talking about further in the class. This is a kind of a slick technique developed by Heng Li at the Broad, and basically what it does is it penalizes the base qualities in alignments where a read is close to an insertion deletion polymorphism. So again, it's addressing the same problem as you see in these realignments around short insertions deletions. Effectively, by decreasing the base quality in the raw reads close to those indels, you're less likely to infer a SNP because the qualities of non-reference alleles will be lower because they're adjusted by this BAQ or base alignment quality adjustment technique. So this is something we incorporated in our own pipelines. We generally ignore reads in the SNP calling if they have evidence of being multiply mapped. So this is where the read quality is 0 in the BAM file, which I told you about. If the mapping quality is 0, then that's an indication that it's multiply mapped. And then these are downstream steps that after you've called SNPs, okay, in your data set, then you can begin to filter SNPs, again, using this sort of arbitrary technique of deciding on thresholds where you identify some criterion and say, okay, anything that meets that criterion, we're going to get rid of it. So one criterion might be to use a program called Repeat Masker to identify repetitive regions in the reference genome, and then just simply not call SNPs in the repetitive regions, because repetitive regions are notoriously difficult to sequence and enriched for false positives in the SNP calling sets. We can filter SNPs with anomalously high read depth. So one of the signatures here of this pattern here, how do we detect this? How do we even begin to address this problem? Well, one thing is to consider is that the sample genome, for a duplicated region that maps to a single region in the reference, you should have twice as many reads as your sort of null expectation for what should happen in this scenario, compared to regions where the sample is not duplicated relative to the reference. And so this leads to this idea that maybe we can, get rid of most of these cases simply by, filtering on very high read depth. Okay, so that is a common filter that's applied. We can filter variants with heterozygous genotypes in diploid calls from haploid samples. Okay, this is something that I've talked about in other lectures that I've given on the subject, but I won't go into any detail here. Other than that, if you do a diploid, if you call SNPs in a diploid using a diploid SNP caller, and yet you're working with a haploid sample, say where there aren't alleles from both parents, if you find heterozygotes, those are unexpected, and so this is a way of filtering in haploid samples anyways. So imagine you could do this for, say, the X chromosome in males, where you have an X and a Y. In male humans, this might be a technique you can adopt, because the X and Y are not homologous chromosomes. You can filter variants with excess heterozygosity relative to the expectation from the allele frequency. This incorporates population genetic data to sort of deal with this, with problems where you see way too many heterozygotes sort of at a locus than you would expect by chance. Again, some of these are, I don't want to go, I don't want to lose some of you here. I want to introduce as much as I can, some of the details and some of the complexities in the SNP calling process. But in the end, most of this, again, is just a few command lines in a pipeline that are meant to address some of the most egregious known problems with the resequencing process. And none of this do I expect you to memorize. It's more just to begin to familiarize you with some of the problems with our field and the analysis sort of difficulties that those of us who are working with the raw data are trying our best to get, as high quality, or to generate the high quality SNP data sets that allow us to draw conclusions about the biology or whatever question we're trying to address. So I just thought I'd mention here, the previous slide was, and that section was all about mapping error, and I told you about how mapping error is a significant problem in next generation sequence analysis. Another problem is in the genotyping step in inferring whether an individual is a homozygote or a heterozygote. And this problem has a couple of sources, the basis for the problem is that this alignment bias problem, which I mentioned a moment ago. Once again, this is where, imagine an allele in a sample that you've sequenced. So imagine the sample is heterozygous at a particular locus, and one allele is a perfect match to the reference, whereas the other allele has multiple mutations in it relative to the reference, and therefore the probability of it mapping correctly is lower. And so one problem here is that you end up enriching, your alignment for the allele that is identical to the reference, and what that does is it makes that site look more like a homozygote, for the reference allele than its actual true status as a heterozygote. I hope that's clear. But then we have a second problem, which is the allele sampling. So here, remember that humans are diploid, and at heterozygous sites, if you have low coverage, the probability that a read comes is imagine you have, in extreme case, only 1x. at a site. That's one read. So you're only going to be sequencing one allele. You cannot sequence the other parental allele, correct? So in that event, you are, forced to call, you would be forced to, you wouldn't be unable to distinguish a homozygote from a heterozygote. Well, usually we're not dealing with 1x, but imagine we had 4x. So just by chance alone, out of 4 draws from a heterozygote site, so you have a 6.25% chance of not sampling both alleles in a heterozygote sample. And so 6% of the heterozygous genotypes in a 4x coverage genome will not contain the signature of a heterozygote. They will not contain both bases at a position that is heterozygous in the genome, simply because of what we call binomial sampling. So drawing these samples where you have a 50% chance each draw, okay, where each read represents a draw or a random sample of that site, 6.25% of the time at 4x coverage, we will not sample both alleles. So we're concerned about this in some of our data sets. One of the projects I'm working on is working on the date palm genome, where we're sequencing 100 different varieties of date palm. This is in collaboration with New York University, Abu Dhabi, where I spent some of my time. And we're really concerned about this problem because of how heterozygous date palms are. There are millions of positions in the date palm genome in any one individual date palm variety that are heterozygous, and we're concerned that we're going to really be missing a huge number, the raw number of sites that will be misgenotyping due to this type of problem is significant, particularly in regions that have low coverage. So this is just a slide I threw in to answer how significant that problem is. I won't go into any additional detail there. So one of the things I want you to take away from this lecture is that in the absence of having these high quality benchmark data sets like they have in the SNP, like they have in humans, such as the HapMap data sets, we're left with this strategy where, in order to reduce the false positive rate, that we need to filter our data. We all agree on that. The question is how to filter it. And so here what I've done is I've just gone to a number of very prominent papers, and I just went into their methods and I looked to see, well, what filters do they apply to reduce the false positives in their data set, which is generally the goal. We usually want to eliminate as many as we can, and yet not, and still retain, the vast majority of SNPs that are real. So this is some SNP filters that the people working on malaria used in this recent Nature paper. They simply removed all SNPs that were away from genes, that were not in genes themselves, and the reason was because their mapping rates, they had a lot of mapping errors outside of the genes because of lots of insertions and deletions occur outside of genes, and in the end that translates into more mapping errors, and which translates into more SNP calling errors. So they simply just didn't analyze data outside of genic regions. They removed reads that were at low, or SNPs that were at low population frequency, because those tend to be enriched for false positives. They required a minimum depth of coverage, okay, where at least one sample, I think they had 200 or some samples, at least one sample had to have 10x or greater coverage at a particular SNP site in order for them to retain that variant in their data set. So biallelic, so you can also have triallelic SNPs, so that is where you have at a particular position within a population, you can have 3 alleles, in at least one individual somewhere in the population. Obviously, in a diploid case, you can only have 2 alleles in any one individual, but in an entire population, you can have 2 or 3 or even 4 alleles representing the 4 nucleotide bases at any one position, although biallelic are by far the most common. So anyway, these are some examples. This was from a sorghum. This is a type of grain. that's typically used for cattle feed around the world, where people went in and these are the qualities that they required in order to retain a SNP. So the SNP quality or the variant quality that I've mentioned had to be greater than 20. So if you remember on a Phred scale, that's the error probability has to be less than 1 in 100. That's what a Phred quality of greater than 20 means. They removed regions that had a lot of SNPs in them. So if you had more than one SNP within a 5 base pair interval, then that interval was excluded, et cetera, et cetera. So people are using these filtering strategies because they suspect that these filters are having dramatic problems or introducing problems in their SNP calling. Here's another example. Feel free to have a look through these and ask questions, but typically what you see is people are filtering on variant quality. They filter individual samples based upon genotype quality. They also filter based upon overall variant depth, so that would be summing all of the reads across a population of samples, or depth at individual genotypes, as we saw in the Plasmodium case, where at least one individual had to have a depth of 10 within that individual samples of reads. So the thing is that these are somewhat arbitrary, many of them. So what can we use then to actually inform ourselves about what classes of SNPs, like low coverage SNPs that have very low coverage across a set of samples, or SNPs that are in repetitive regions, et cetera. How do we know which of these sets of SNPs are really contributing a lot of false positives? Well, I alluded to this in one of your homeworks where we talked about the transition transversion ratio, and I won't go into it into any more detail here other than to say that this is the one criterion that if you do not have a benchmark data set and you're working on a non-model species like date palm, like I am, or on Chlamydomonas, the algae, like I am, we have to have ways of identifying classes of SNPs that are false positives that are likely to be artifacts of one sort or another. And the only thing that we have in these types of data to rely on is the transition transversion ratio. And the basic principle is that the transition transversion ratio, these are classes of mutation. So A to G or G to A, that's a transition, and C to T or T to C, that's a transition. So the black arrows here are the transition steps of mutation, and the blue arrows represent mutations that introduce what we call transversions. And the point is that there are half as many black lines as there are blue lines in this slide, okay? And that means that by random chance alone, if we introduce a random error in our data set, that more often than not it's going to represent a blue, twice as often it should represent one of the blue changes compared to the black changes, right? Because there's twice as many possibilities for introducing an errant C at a site that's A in the reference, okay, than there is introducing an errant G at a site that's in the reference. So the blue changes are twice as likely as the black. And so this, whereas in biology, what we observe is that SNPs have the reverse, okay? It's usually 2 to 1. In human exons, it's 3 to 1. So there are more true polymorphisms where at any one position there's an A and G, that's a transition. These are twice as likely as the transversions. Okay, so you're twice as likely to see an AG polymorphism as you are to see an AC polymorphism. Okay, so this gives us a way of identifying classes of SNPs. So what I do is I say, okay, I'm going to look at all variants where the quality is a Phred score of less than 20. And what I would expect to see is that the transition transversion ratio will be lower in that class of sets of SNPs than a higher quality set, say a set with a Phred quality of 30, which is a 1 in 1,000 error probability, compared to a Phred of 20, which is an error probability of 1 in 100. Okay, so the higher error probability set, okay, the set where we're more likely to see errors, we would expect to see the transition-transversion ratio to be lower. And in fact, that's what we see, and that's a fairly obvious case, but in other cases, we use this as a guide to identify classes of SNPs that have particularly low transition-transversion ratios and are therefore likely to be enriched for false positives. Okay, so let me just offer a word of encouragement here to you. This is a topic that I've spent 2 years working on. It's a subject that we worry about in our data because we're worried about having high quality data. For somebody in your position where we're just trying to introduce you to the process of SNP calling and how to get high quality call sets, you now have a resource to go back and to review this material as you become an expert in next generation sequence data analysis. What I expect for you to be able to do in this class is to execute the series of commands that will, allow you, you to come up with a reasonable call set of SNPs, and to be aware of the potential issues that exist that are out there, and to have some sense that there are issues out there. They're understood. What those issues might be, but in the case of many of the particularly non-human data sets, we have issues with identifying the sources of error and the problems with these call sets. And what I'm showing you here are methodologies that people working on non-model systems frequently revert to. Human data sets, you see filtering steps as well. People have developed sort of set protocols for how you can filter, in addition to doing those more complicated things like base quality recalibration and variant quality recalibration. But in any event, the filtering step is critical to getting high quality data, and hopefully the last few minutes of lecture have shown you some of the tricks that we're using and how variable it is for what filters different people apply to the data are largely specific to those data sets. So this is a totally different topic now. We're almost to the end of the lecture. This is a long one, and I apologize for that. You feel free to, hopefully you've, had put this on pause and, had something to drink or whatever. But what I'd like to, just in the last couple of minutes here, is introduce you to the functional annotation step where we deal with SNPs. I'd like to tell you about the VCF format briefly, and I'd like to introduce you to the GATK. So what I'm going to do is I'm actually going to pause the lecture here, and I will introduce these final concepts in a second lecture. So we'll take a break now, and so head back to the Blackboard site to get the link for the second part of the Week 6 lecture series. Okay, talk to you soon.